Ja, sehr verehrte Damen und Herren, dear ladies and gentlemen, my name is Ulman Lindenberger, and it is my great pleasure and great honor as one of the vice presidents of the Max-Planck Society and on behalf of Martin Stratmann, the president of the Max-Planck Society, who couldn't come to Berlin so quickly, to congratulate Emmanuelle Charpentier for the 2020 Nobel Prize in Chemistry for her groundbreaking work on CRISPR-Cas9, a tool for editing the genome. This is a fantastic moment for the Max-Planck Society, and we are very proud that you are one of our members.
Please come closer. Mm-hmm.
Dankeschön.
Can you take it off?
Den Strauß übergeben, okay? With the mask this time.
Okay. Okay.
You're welcome.
Okay, more pictures then? I can take it away.
Thank you very much.
So. So I think I just open and we proceed with the press conference.
Yes, surely.
Because I know a lot of colleagues in the room are in a hurry to get the news in the evening news. We will live stream this press conference, so we have questions from people in the room and questions from people which are on the YouTube channel, and I will try to handle both together.
Okay.
But before, I will hand over to Emmanuelle. I'm very glad that you're here with us after this very big day. Everybody waited for the Nobel Prize and expected you to get it, and today that's the day of the days. So I'm very happy you're here, and I guess you have a little statement or a little intro to tell journalists here in the room about.
Yes, I can say a lot when the days are long. I'm surely very honored to receive, I think, what scientists consider the most prestigious prize in science, and to receive it in collaboration with my former collaborator Jennifer Doudna from UC Berkeley. So it's a special day for me, and as you maybe mentioned, I mean, as much as how should I say, as much as it was said by scientists, but also non-scientists, that CRISPR-Cas would be awarded one day the prestigious Nobel Prize. It was not clear who will be awarded as a scientist having been involved in this research, and also when it will be awarded. So this morning when I received the call from Stockholm, I was very, very emotional, I have to say, extremely emotional. And if I can say further statements, I can go ahead by thanking everyone who has been involved in this research. So my former students, specifically Elica Delcheva and Krzysztof Zielinski, back when I was directing a lab at the Max Planck laboratories at the University of Vienna in Austria, and back when I was at the Laboratory for Molecular Infection Medicine Sweden at Umeå University. So this is really where this discovery developed. I want to thank my collaborators. I would like also to thank all the scientists who have been involved very early on in the CRISPR-Cas field of research. And also all the scientists and developers who have been a lot involved after the release of our science paper in 2012 describing these genome editing technologies and describing the guidelines how to use it. We saw a lot of scientists and developers literally jumping on the technology and applying it for their own needs to perform genetics in multiple cells and organisms. And now we really see the development in biotechnology, biomedicine, with even patients being cured thanks to this technology. What I would like to mention, if I can continue, is that it is a very important prize because it awards a field of research that is called fundamental macrobiology, and that is very timely in the time of the pandemic which we are experiencing, reminding everyone that infectious diseases is the number 2 of deaths worldwide in terms of diseases, and that bacterial infections remain a concern, with a lot of clinical isolates still resistant to antibiotics. A lot of research that have been done in the past on bacteria and viruses, but a lot of experts as well who are now retiring, with really the need to bring new scientists to continue this timely research. CRISPR-Cas is about, initially, before having been developed as a technology, is a system that bacteria and archaea, so macroorganisms, have evolved to allow them to develop an immunity against their own viruses. So I think now people understand what it means. So bacteria, archaea, like us, can be infected by viruses and can develop an immune system that allows them to fight and to resist the virus. And CRISPR-Cas is such a mechanism. So I think it maybe says something to the people, but this type of research has really led to this technology, CRISPR-Cas9, that is a technology that allows to perform precise genetics in multiple cells and organisms in an unprecedented manner. But what is really critical to highlight is that it's a very perfect example of a discovery that has a large impact in life sciences, in research and development, but also in biotechnology, in the production of food, in medicine, and that is really originating from pure fundamental research, going very deeply in the mechanisms of life, studying evolution, studying the diversity of microbes, trying to understand how they cause disease, trying to understand how they interact with one another. And this is through this type of research that CRISPR-Cas9 emerged. And this is the reason why the prize is awarded today.
So, waiting for the questions? So we're waiting. Do you have any questions? Yes, yeah.
My name is Nils Schneider. I work for German microphone.
Yes, we have the microphone in the middle of...
Nils Schneider, German Television, ZDF. First of all, congratulations to this wonderful prize. You said you received the call this morning. I have 2 questions. Could you tell us where were you, how did you react? And second question, how does this prize affect your work now? Will something change?
So for the first question, I was sitting at my desk. It's a working day. And so I was sitting at my desk working, and I received a call that was clearly from Sweden. And so I took the call and I received the news. Actually, I was the first one they were calling because they had my cell phone number. And they asked me actually to provide the cell phone number of Jennifer Doudna because they did not have it. So this is for the little story. And yes, surely I was very emotional because, it's one thing to be told multiple times that maybe one day you would be awarded the Nobel Prize, but another thing when it happens, whether it will, what will it change in the future? It will not change me. The people who know me know that I will remain the same person that I have remained all over the years. What will not change, but what will, I think, helped me, I believe, is to provide the message to the non-scientists and the public or people who may not be so familiar with science, that fundamental research is critical, and it's critical to be supported in our days. The concern that the upcoming generation may not continue in the academic field, maybe for justified reasons, it's not easy. That this field of research, which I think now people understand that is critical, infectious diseases, microbiology, it's really a field that is critical to be supported highly. And this goes beyond the development of a vaccine. This goes with making sure that experts are still trained and present, who know very well how to do research with multiple bacteria and viruses causing diseases, not only for humans, but also for plants and other types of organisms. So I hope that this prize will help me to highlight this message, and also to highlight the need to support academic science. It's not only supporting scientists. Scientists are supported if you support well the system around the scientists. It's a huge machinery involving, a number of people who are not the scientists, but who are helping the scientists to be able to do their science. And in the last years, the scientists have suffered from the fact that they do everything else but science, at least at leading functions. And that it's really a huge machinery involving foundations, organization, taxpayer, money, and that a lot of people work very hard and are also the non-scientists very enthusiastic in working in scientific organizations. And maybe with what we are witnessing and what we are experiencing now with the pandemic, I hope that it highlights in every one of us this interest in science. CRISPR is also what it's changing. It's maybe the fact that it's more visible, so it allows to understand that innovation technology also always involve fundamental science. It also allows me to highlight, because this is a case, that it's not about doing science for publishing for the sake of publishing and accumulating impact factors that are the way scientists are judged or evaluated. It's about believing in a field of research, a topic, a project that may not be that fashionable or exciting at first, but that has potential and that involves time, focus, going deep in the research, solid work, at the risk of not publishing for a while, but having, bringing the effort and the commitment to do as much as possible research that can have value for humanity and society.
Mm. Thank you.
The prize also allows me to highlight the fact that 2 women received the prize. I consider myself as a scientist, but I was asked this morning this question, and that's why I say it. I want to mention the fact that you have a lot of prizes, Nobel Prizes, that are awarded maybe 20, 30 years after the discovery. This prize is awarded 8 years after the discovery. And what it does by awarding it to 2 female scientists, leader scientists, is just highlighting what is happening in our days. Science, fundamental science, becomes slowly, but hopefully surely, let's say a word or so of female scientists as leaders. And it reflects what is happening in our days. Work often involving collaborations, and collaboration that may be collaborations among male scientists, among male and female scientists, or among female scientists, and this is what is highlighted through this prize, and I hope that it also provides an image of the science that is more modern, whereby also the girls and young female scientists see understand that nothing is impossible, and they can also go ahead with their career and have themselves find great discoveries, have great findings that can be impactful. It's independent of the gender.
Okay. Some more questions from the floor here in the room? So they came in some questions over the chat. One question is a totally different direction. One person asks, how did you react when you just learned that you won the prize?
And
it's if you could answer in French, that would be fine.
If I could answer in French, my God. Je vais répondre en français. J'ai visiblement reçu beaucoup de prix. Celui-ci est très particulier puisqu'il est considéré comme l'ultimate distinction de tous les prix qu'un scientifique ou qu'une scientifique puisse recevoir. Même si, et je pense que c'est de notion commune, qu'un jour un prix Nobel serait donné à la découverte, si on peut appeler ça comme cela, CRISPR-Cas. Ce matin, j'étais très très émotionnelle. Je connais la personne en fait qui m'a appelée, puisque je suis membre de l'Académie royale de Suède de sciences. Et j'étais très très émue, oui, en effet, très très émue. Et je le suis encore.
Okay. Another question, maybe you can give a little summary of this in English also, because I think a lot of people are interested in the answer.
Yes. So again, I'm blunt. It's been years that people think that one day CRISPR-Cas will be awarded the Nobel Prize, and that maybe I will be one of the awardees. But I was very, very emotional when I received the call from the Royal Swedish Academy of Sciences. I was very emotional. For sure, it means a lot to me. A lot of things have happened in my life the last 12 years. It has been a marathon. And so many great developments about this technology. It's quite amazing. And a field of fundamental research around the CRISPR-Cas field that has been also amazing. The last 10 years I've witnessed so many publications in the field, the description of so many new mechanisms and great applications, and enthusiasm of scientists also means to understand that unfortunately not in Europe, but in the US, a great scene with regard to biotechnologies, which are also critical in pushing forward through the funds they can gather, pushing forward great applications of the technology.
Okay. Another question about Berlin. How long do you live in Berlin, and which district, and what do you like about Berlin, and how are you going to celebrate the award today?
So I've been living in Berlin for 5 years. I wish I would have been able to give this press conference in German. I'm living in the middle of the city. Maybe not the great place to appreciate Berlin. It's maybe too much gentrified, but I take the time when I have the time to experience other areas that are very interesting, because for sure Berlin is a very exciting city, specifically with regard to the history and it's like CRISPR-Cas. But it's not like CRISPR-Cas because it's always under development, right? You may have realized it, but it does not go as fast as the development of CRISPR-Cas. What was the next question?
Oh, how you are going to celebrate?
As a matter of fact, I was invited for dinner this evening. I wonder whether the date was chosen on purpose or not. by the former Senator of Science of Berlin, Jürgen Zollner, whom I say hi if he's listening to me. So maybe at his place, then I will have to make it short because I have to reply to a lot of questions. Jürgen Zollner was being very at the political level because, you know, politics is also extremely important for academic research. This is just a fact, and I just say it because we need the support of the politicians to continue our fundamental research. But Jürgen Zollner has been very helpful with regard to welcoming me in Berlin.
Okay. Another question more about your methodology. Is CRISPR-Cas also is it possible to use it against COVID-19 to develop something?
So it's used actually. There are some kits which have been released with regard to using the CRISPR technology to help the detection of the virus. Now directly using CRISPR-Cas as a kind of vaccine against COVID, I see it very difficult to imagine that it could be used directly. But indirectly, surely, because one has to understand that CRISPR is a technology that is really used by biologists and developers to help finding new targets for therapeutics with regard to SARS-CoV-2. This is what we are doing also in our lab, conducting CRISPR screens in human epithelial cells in conditions of infections with a virus to try to find new components of the human cells that are interacting with the virus and to try to understand better how the human cells, what are the mechanisms involved that allow to understand how our cells interact with the virus. So indirectly, the technology has a use. One has to understand that prior to CRISPR-Cas9 and prior to two other types of nucleases that were in place prior to CRISPR-Cas9, but that were not really used, you know, in let's say democratic fashion. CRISPR-Cas allows to really perform genetics in human cells, and it's a very extremely important time in the context of human diseases, where one wants to be able in the lab to study, in the case of our diseases, the interaction of the virus with the human cells. If you cannot after do genetics in human cells, you don't go very far. So CRISPR-Cas will have a huge impact in this regard.
Okay. Questions from the floor? New questions? No? So I just proceed with the chat questions. Somebody's asking about the ethical responsibility because there are a lot of powerful genome editing tools which you added to science now. And some scientists think that your discovery may be diverted for less positive goals on human genome. What do you recommend to avoid this?
Yeah, so first of all, I have to say that with regard to the media, I like very much the fact that the media have highlighted very early on as you know, being a transmitter of the communication between the scientists and the public, that CRISPR-Cas9 was a technology with numerous positive effects. And this was very important to convey first this message. Now, for sure, you have ethical implications, and that with a number of national academies that have released statements with regard to their, their position with regard to the ethical application. Actually, Leopoldina, the National Academy of Sciences in Germany, was, I think, the first one to release a statement. So, in a nutshell, the technology surely could be used for what we call human enhancement, so for designing babies with certain genetic traits. I think the scientific community as a whole is surely raising itself against the usage of this technology on this aspect, in the sense that for the use in humans, it is clear that the technology is meant to be developed to cure diseases, or in the context of diseases with a genetic background, with mutations, but not for human enhancement. in any case, the technology is not ready. You need, or maybe it is relatively clear that it is like a medicine. Before a medicine reaches the market, you have a lot of checkpoints. If you take the CRISPR-Cas9 technology up to now, it's not yet not ready in any case for performing genome editing in a precise and safe manner in human germlines. And even if it was, you have a lot of characteristics that are, plurigenic. It's not one gene, but multiple genes involved. And often we don't know of the complexity of the genes involved in a particular trait. So it's a risk, but it's also, let's say, for maybe certain applications which some people will think it should be applied, I will think not ready in any case nowadays. But technologies go very fast. That is why, it was important to release statements to go beyond if you wish, the development of the technology and have clear statements before the technology reaches this point.
Okay, thank you. I think we have to watch the time. You're in a hurry. So half an hour is over with delay. Thank you. So this is the end of the press conference.
Okay, good.
And
excellent.
So then there are some.
Yes.